So today I'm going to discuss Alzheimer's disease, specifically a paper recently published out of Nature titled Lymphoid Gene Expression Supports Neuroprotective Microglia Function. And a core functionality of microglia in AD progression would be to, number one, regulate the density of these plaques as well as the phagocytosis of these plaques. And remember, that's just an ideal, and physiological systems aren't ideal because things go awry. And what this paper did was it explored the conjecture that neuroprotective states are regulated by the expression of PU1. PU1 is a protein found at various concentrations in microglia, though it functions as a pioneer transcription factor and itself is a master regulator. And recall from our study of molecular biology, a pioneer transcription factor functions, which completely blew me away when I first found out about this, by the way. It makes genes that were previously inaccessible accessible, allowing us to express proteins in this given cell that were previously not found, conferring new functionality to this cell. Okay, and likewise, PU1 is a master regulator, so it is capable of regulating its own expression. Alright, and this could be either directly or indirectly, so it pretty much produces something that later could upregulate its expression itself. Alright, this is important, say, in transmitting cell memory so that we could have cell differentiation. And this cell differentiation is important for having, say, hepatocytes, okay, enterocytes. We want these cells to remain specialized. We don't want them to perform other functions because this could cause abnormalities. Okay, and the importance of PU1 being expressed at a low concentration and functioning as a master regulator, PU1 is able to induce a lymphoid lineage differentiation. Alright, but the caveat is that this has to be expressed at a low concentration. Alright, so we have to find PU1 at low concentrations for this to occur. And this paper explored this nuance fairly extensively. And this nuance was targeted in the study because previous studies actually correlated a low PU1 concentration with later Alzheimer's onset and decreased severity. That said, microglia, which are the primary immune cells of the central nervous system, can be either harmful or beneficial depending on the expressivity of SPI1. And SPI1 is the gene that codes for PU1. Okay, so the protein that results from the gene. And that was shown in this paper, which I will discuss extensively via CD22, which is pro-inflammatory, and CD28, okay, a T cell co-stimulator protein, is actually responsible for regulating the aggregation of these plaques, as its knockout demonstrated a remarkable increase in the plaque aggregation, and it was inversely correlated with the expression of interferons, observed in this paper to be overall beneficial to the organism. Though more on CD28 later, okay? So to summarize this paper, before I discuss it quite thoroughly, I'm going to say that largely, this study collected data supporting the hypothesis that decreased expression of PU1 is correlated with longevity, demonstrated in this study using 5x FAD mice, a common transgenic mouse model emulating Alzheimer's disease in humans. And this hypothesis was further supported, observed in the post-hoc analysis. Decreased SPI1, which codes for PU1, the protein, so a low cerebral PU1 expression, in other words, in microglia, is associated with neuroprotection by impeding Alzheimer's disease development. And this was gauged through the observation of plaque aggregates. And number two, that specifically CD28, the lymphoid gene of interest, likewise confers neuroprotection, observing that, generally speaking, mice with, or may I conjecture, patients with increased CD28 are more neuroprotected from AD progression, which the expression of PU1 and CD28 are intimately related, because we see that when PU1 expression is decreased, CD28 is increased. Thus, this finding illuminates a novel avenue to treating Alzheimer's disease via an antibody-based microglia-driven immunotherapy, which we will discuss later. Alright, now we are going to dive a little bit deeper, okay? So, this study performed a series of experiments to validate and rule out a number of confounding variables that may have arisen between experiments. To begin, This study investigated the AD-prone mice model, that 5X FADA model. We understand exhibits a genetic profile analogous to that observed in Alzheimer's disease. In this paper, discovered a subpopulation of microglia among the DAM, so the disease-associated microglia, showing decreased levels of PU1, abbreviated as PU1 low, in contrast to PU1 medium and PU1 high. This paper, however, focused on PU1 high and PU1 low. And they show vast differences in gene expression. Once again, this paper emphasized the juxtaposition between PU1 low and PU1 high. Interestingly, PU1 low preferentially co-localize with amyloid plaques. And this is observed in both the 5X FADA mouse model as well as in humans, which ultimately these DAM microglia increase in number as the disease progresses. So we would expect to see a high concentration of PU1 low microglia in populations of really sick patients with Alzheimer's disease. That said, PU1 high does not express this selectivity. So it's not going to preferentially co-localize with these plaques. Alright, moreover, CSF1R, which is a specific receptor, once postulated as the essential microglia receptor, so their existence would depend on this receptor, does not appear to be implicated in the development of these PU1 low microglia. This lack of dependence was investigated by inhibiting the CSF1R receptor, observing that the non-plaque associated microglia died off, whereas the PU1 low disease associated microglia, so the microglia associated with these plaques did indeed survive, displaying an exception to the essential microglia hypothesis, thus causing us to impugn the origin of these microglia, so these PU1 microglia, to see if they really are authentic bona fide microglia. However, lineage analyses were indeed performed, confirming that these do have an authentic microglia origination, so that just rules that out. So these are, yes, microglia, showing an exception to the essential receptor hypothesis. So diving into PU1 itself, so the protein that results from, recall, the SPI1 gene. It's observed that cells naturally downregulate SPI1, so we observe less PU1 in response to plaque-associated cues, so in response to the formation or observance of these plaques, or through the recruitment of a PU1 low microglia subpopulation to the plaque themselves, though this study focused on the latter, so just the recruitment. Alright, so to discuss the nerd stuff, the biochemical pathway that we are going to observe, membrane-bound receptors are number 1, responsible for the plaque-driven morphological changes, and number 2, the transcriptional responses. So we have, say, a membrane-bound receptor, TREM2, for this example, being bound, then activating tyrosine kinase, which then there being a series of intermediates yielding another secondary messenger of interest, PLC gamma 2. So PLC gamma 2 and tyrosine kinase being abbreviated SYK, are going to be the two messengers pertinent to this discussion. It's observed that the inactivation of SYK, so tyrosine kinase, or PLC gamma 2, both severely impaired microglia plaque associations. So decreasing SYK, tyrosine kinase, is associated with demyelination, so decreased long-term potentiation, so impaired memory, so that may account for the memory deficits, as well as uncontrolled plaque aggregation, and the sequential knockout of SYK and PLC gamma 2. Number 1, ruled out any potential confounding variables. Number 2, the SYK knockout confirmed that PLC gamma 2 is in fact a downstream component of this pathway, resulting only from the upstream tyrosine kinase. And number 3, supporting the importance of PLC gamma 2. However, the mechanism was not delineated in this study, unfortunately. Likewise, the inactivation of both SYK and PLC gamma 2 results in decreased PU1 low microglia, which are the plaque-associated microglia. That said, we can deduce that this SYK PLC gamma 2 pathway is critical for PU1 low microglia, which this pathway accomplishes theoretically by downregulating the SP1L gene. Okay, so moving on, we're going to discuss the liposomal and beta-glucan experiment. Ex vivo, so outside of the organism, microglia were exposed to phospholipids, okay, specifically phosphatidylcholine and phosphatidylserine, commonly observed in biological membranes, and beta-glucan, specifically beta-1,6-pustolan, I apologize if I butchered that name, which engaged TREM2 and CLE7A receptors respectively. And we recall that the TREM2 and CLE7A receptors stimulate SYK and the downstream POC gamma 2. A set of experiments collected evidence that these two particular pathways are responsible for regulating, either directly or indirectly, PU1 levels, further substantiating POC gamma 2's role in decreasing PU1 levels. Alright, to focus on the liposome experiment, we observed that increase in the concentration of phospholipids appears to remarkably decrease PU1. However, these effects are in fact reversible as observed by a follow-up washout, okay? Likewise, the beta-glucan downregulated PU1, which this makes sense because the earlier supposition we made claimed that POC gamma 2 is responsible for decreasing PU1, and if these two pathways are responsible for upregulating the SYK POC gamma 2 pathway, thereby increasing POC gamma 2, we should observe that the increased POC gamma 2 is in fact contributing to lower PU1 levels. That said, a subsequent related experiment actually inhibited POC, and guess what we observed? We observed normal PU1 levels, which confirms the role of POC gamma 2. Alright, so now we are going to actually move on to like the meat and potatoes of this paper. So the association of decreased PU1, okay, so PU1 low being associated with lymphoid gene expression. Okay, so PU1 low microglia being associated with lipid metabolism and lysosomal function show a gene expression pattern similar to lymphoid lineage cells, specifically T cells. Of interest is CD28, a co-stimulatory receptor commonly expressed on T cells, but in this case we see it on a microglia, specifically PU1 low microglia, not PU1 high microglia. Most relevant is that an analogous expression is realized on human disease-associated microglia, so we also see this lymphoid lineage gene expression. And I only mention that because this study emphasizes 5x FAD mice, okay, so the mice that emulates AD, but because we are humans, we often care about what pertains humans, alright? And yes, this model closely emulates AD, but it becomes increasingly compelling because we observe this similar gene expression in both humans and mice. Okay, besides that note, it was determined experimentally that decreasing PU1 levels postnatally, so a SPI1 gene knockout via CRE recombinases, resulted in lymphoid-related ontologies. Conversely, when increasing PU1 through the addition of an additional allele, so increasing the gene expression, they observed a pro-inflammatory pattern. And most importantly, these findings were actually indicated in the absence of plaque, illuminating a potential clinical preventative approach, essentially pre-programming microglia to be prepared for plaque aggregates before they manifest. Alright, so a PCA analysis illustrates that PU1 low microglia assume a similar gene expression pattern to T cells. Okay, and this was observed to be an ATAC sequencing, a technique assessing genome-wide chromatin accessibility. Recalling from earlier that PU1 is a pioneer transcription factor, so it's capable of making previously inaccessible heterochromatin accessible, more like euchromatin, essentially conferring the microglia more tools in the toolbox to actually combating such aggregates when they do manifest. Otherwise, I want to emphasize briefly the importance of heterochromatin. Okay, so we don't want parietal cell enzymes, for example, to be found in our eyes. That's going to be localized elsewhere. Okay, so we want cell specificity to remain intact, alright, and we want differentiation to occur appropriately. However, pioneer transcription factors are actually able to, in response to different signals, tamper with this process, essentially. So really, to tie things together and make it more comprehensible, as SYK, okay, tyrosine kinase, PLC gamma 2 pathway signaling increases, we observe a decrease in PU1 levels, and the PU1 low microglia experience a change in their protein arsenal, essentially, by opening previously inaccessible heterochromatin, that which is observed in T cells. Okay, so it's going to open, make accessible genes that are exclusively, typically, observed in T cells, alright? Okay, and this elaboration is confirmed by PC1, ensuring to notice that the PU1 low microglia is not superimposed on the T cell region, as that would indicate uniform expressivity, and if that were the case, this PU1 low microglia would itself be considered a T cell. The point of this elaboration is to accentuate the point that such PU1 low microglia adopt a gene profile similar to T cells, and that this is not observed in PU1 high, more so adopting a genetic profile conducive to inflammation. Specifically concerning microglia with high levels of PU1, we observe increased levels of interferons, and number 2, C1q complement activation. And for our purposes, we are concerned with either the inflammatory or phagocytic complement pathway, and it appears, this is conjecture, that the inflammatory pathway is activated by C1q. Conversely, we see that microglia with low levels of PU1 exhibit behavior that remarkably stifles AD progression, and this is accomplished by enhancing microglia plaque interactions. Alright, so I keep talking about CD28, but I think it's appropriate to just divulge on that a little bit. It's a co-receptor / co-stimulator, earlier explored in helper T cells, which is activated by binding to a ligand, an epitope on a foreign molecule, then activating the actual T cell itself to perform a job. Recall from molecular biology that microglia itself can interact with the molecule, say for our purposes an amyloid plaque, resulting in activation of a single pathway, say in our pathway a phagocytic pathway. However, the expression of CD28, a T cell receptor, on this microglia will itself activate a discrete signal, initiating a pathway either independent or related to the pre-existing set pathway, though my discussion on this actual pathway is only conjecture because it has not been explored in the literature as of yet. As I can only imagine, that CD28 associates with the microglia membrane, either adjacent to or quite the distance from the microglia's primary paratope. Akin to how two different arms can interact with a given entity from two different angles with varying approaches and causing different effects based on different interactions. In other words, immunology is very complex, though I am excited to observe the topology revealed in future studies. So let's discuss CD28. Number 1, we observe that CD28 lymphoid disease-associated microglia increased by almost 4x, and number 2, and almost 2x in the number of plaque-associated microglia. Additionally, the fraction of CD28 microglia increased, whereas the levels of MX1, a type 1 interferon, frequently associated with neurotoxicity, decreased. In addition, there was an observation of an increase in the plaque density, alright, so a reduction in this overall surface area of plaque, showing that low levels of PU1 are responsible for a sort of plaque-associated quality control. Okay, so this increase in plaque density actually impairs the capacity for it to spread for reasons that I will discuss subsequently concerning axonal integrity. All in all, I could continue on with my infatuation of PU1 low microglia and their positive effects. However, these are all positive once again, and I don't suspect that these researchers were approaching this with the intent of looking for any negatives. So though I didn't mention any negatives explicitly, just know that they may be lurking somewhere. Okay, and the working hypothesis that CD28 is number 1 responsible for decreasing plaque propagation and number 2 for neuroprotective effects was supported by several experiments, which I will discuss the results to those experiments now. Okay, so experiment number 1 showed that decreasing CD28 through a knockout actually increases the plaque surface area, thereby showing that once again CD28 is responsible for like a quality control mechanism, increasing the density of the plaques. Okay, and during the second experiment, as shown by the UMAP dimensionality reduction, CD28 knockout displayed a remarkable increase in type 1 interferon expression. In experiment number 3, we observed that a CD28 knockout had no effect on the total number of microglia. However, the volume of the plaque was significantly greater. Recalling once again that CD28 is responsible for quality control. And the last results I will discuss concerning CD28 functionality demonstrated that a CD28 knockout resulted in an immediate increase in MX1 expression, a type 1 interferon. As discussed earlier, MX1 is implicated in neuroinflammatory pathways, further suggesting that CD28 is in fact responsible for reducing neuroinflammation. And this is all in comparison to CD22, and this is upregulated in PU1 high microglia, is ultimately associated with decreased phagocytosis, and we want to be phagocytosing these plaques to ultimately decrease the number of plaques. CD22 associated with PU1 high, so high levels of PU1 is associated with decreased phagocytosis, so the decreased ingestion of these amyloid plaques, which is ultimately unfavorable for the progression of Alzheimer's disease, because if we are experiencing an increase in the number of plaques, we want to have a mechanism to get rid of those plaques, right? So we don't want to have decreased phagocytic activity. So now I'm going to discuss the tangible benefits of PU1 being expressed at low levels. Though first I'm going to discuss common observations of 5x FADA mice. So 5x FADA mice, so the Alzheimer's disease mouse model, are observed to have decreased presynaptic terminals, impaired synaptic plasticity, so a decrease in long-term potentiation in the hippocampus specifically. So this is crucial in memory, by the way, and other psychiatric maladies concerning object recognition tasks, as well as behavioral deficits. That said, when low levels of PU1 were experimentally induced, that essentially did away with all of these negative alterations from manifesting, preserving phenotypes more proximate to the wild type. Wild type referring, of course, to ordinary mice models, not the 5x FADA mice. Even more, low levels of PU1 significantly increase the lifespan, and this in itself gives me a reason to continue exploring the literature concerning PU1, because its association with longevity is increasingly profound. So we have these membrane-bound receptors. So we have membrane-bound receptors bound within the membrane of microglia, which are responsible for regulating the levels of PU1. Remembering that the associated pathway includes tyrosine kinase, so SYK, and PLC gamma 2, which are ultimately responsible for regulating the levels of PU1. And PU1, the master regulator of interest. And this is because low levels of PU1 are associated with CD28, whereas high levels are associated with CD22, which CD28 with the lymphoid cell lineage is speculated to be implicated in reducing neuroinflammation and reducing the surface area of plaque by increasing the density of the plaque. And that hypothesis is supported by the plethora of experiments discussed. And to relate this to previous studies, we have TIM3 deficiency that we're going to discuss. The researchers suspect that CD28 and the associated lymphoid gene expression may lie at the crux of the decreased neuroinflammation and the increased phagocytic activity observed in TIM3 deficient patients. Overall, this paper not only ascertained a novel therapeutic approach to treating Alzheimer's disease, but it also worked to bridge pre-existing chasms found throughout the literature. So that's all I wanted to discuss in the video. I hope you walk away with the consideration of the plausibility of this novel therapeutic approach.